Well, good morning. It's a pleasure to be here. The only thing I really have to disclose is that when Mark asked me to come speak at this session, I told him I didn't know anything about wounds. And he told me if I said the word wound 3 times, I'd be okay. And there's 2. I'm not going to cheat and read the title because that would be too easy. But nonetheless, what I want to do today is present some of the work that we're doing related to genetics and biochemistry, and we'll see if we can't tie some of this together. On a more serious note, my real disclosures are that I work for the government, which means that I literally am not allowed to have anything to disclose. So it was about 10 or 12 years ago that NASA identified some astronauts as developing ocular pathologies. These were retinal pathologies. There's some nuance to it, but things like choroidal folds, optic disc edema, cotton wool spots, globe flattening, and literally changes in refraction. People that went up with perfect vision and came back and needed glasses, and now 4, 5, 6 years later still do. And this went from us not knowing it was a problem to being one of NASA's more serious concerns as we start to look to go beyond low Earth orbit and send crews ultimately off to Mars. When this happened, we had spent a few years collecting data on space station astronauts, and we went to the docs and we said, look, we've got a lot of data that might be able to help. And when we started to dig through that data, what we realized was that there were biochemical changes that we'd observed that helped to differentiate the astronauts that developed these eye problems compared to those that did not. And I'm told this is a very biochemistry interested group, so I wanted to dig deep into the biochemistry. This is a biochemical pathway that we call 1-carbon metabolism. It's a vitamin-rich pathway. It's involved with folate and vitamin B6 and vitamin B12 and riboflavin and biotin. And this is one of those pathways that when you saw it in school, the question was, is this on the test? And it turns out in real life, this thing is on the test. Okay? So when this came up, we had looked at these data. This is some of the chemicals we measured as part of our experiment. And when we looked at homocysteine in the middle of that pathway, in particular, we weren't seeing much when we looked at the data in toto. That is, it wasn't changing much during flight, and we thought, okay, that's good. We're not seeing folate deficiency during flight, so we can check that box. When we broke the code for those astronauts that developed eye issues compared to those that did not, I can tell you the day, the date, and the hour my colleague Sarah Zwart came in and said, there's something going on with 1-carbon metabolism. And what she meant by that was this, that she'd come up with these data that when we looked at circulating homocysteine concentrations, the astronauts that developed eye problems, shown in the yellow line, had significantly higher concentrations of homocysteine in the blood compared to astronauts that did not develop eye problems, shown here in the white line. And what was perhaps more striking is that those differences existed before flight. I show you the homocysteine data here. We had data on 3 of the other chemicals in this pathway, and we saw the exact same thing. Higher concentrations in the blood of affected astronauts compared to those that were not affected. We ruled out as many things as we could and quickly started to look at the fact that when we look at these metabolites, you need to look at the function of that pathway. You need to look at the enzymes in that pathway, and that starts to get into genetic differences. And the fact that we were seeing biochemistry differences before flight immediately led us to think about the fact that this could be genetically predisposed. When you talk about genetics and enzymes, obviously enzymes are proteins that are synthesized based on the DNA. And when you start to talk about SNPs, as we call them, or single nucleotide polymorphisms, that simply gets at the fact that there are multiple forms of the DNA in the population. Okay, these are not mutations, these are not random, these occur in the population. I'll show you one example, and that is this MTHFR C677T SNP. And MTHFR gets a lot of press because it is the most studied of the polymorphisms in this pathway. The result of this is that when your parents gave you DNA, one of them gave you either a C or a T allele, and the other similarly did. So you end up with either 2 Cs, a C and a T, or two T's. Depending on which pair of alleles you got, you insert a different amino acid into that protein, that enzyme, and the amino acid you got will affect the function of that enzyme. And what we maintain is that if you've got what we call the minor form of that SNP, that enzyme will not work as well, which means that pathway will be slowed down, and theoretically everything upstream of that will build up. And that is one of the possible explanations of why we were seeing higher concentrations of homocysteine in affected astronauts. Now, when this came up, this is a decade ago, literally, it was February 2011, and we went off and wrote a proposal to look at 5 of these SNPs. We picked 5 SNPs that were well documented in the literature to have an effect on homocysteine. We picked 5 SNPs out of probably 500 that we could have in this one carbon pathway. I will tell you that the MTHFR SNP that I mentioned is one of the ones that we looked at, and we didn't find anything that I would call exciting there. So while there's a lot of attention on MTHFR, depending on what your answer is for that SNP, I wouldn't take that as a win or a loss in the big picture. The 2 SNPs at the bottom that I'm going to show you more data on are the SHMT1, one of the SNPs in that enzyme, and the MTRR A66G. Those are the 2 SNPs that I'm going to drill in on next. We went off and recruited astronauts, and what they did was literally every single astronaut that we talked to, that we asked for blood to look at their genetic data, said yes. And when we boiled the data down, what we found when we look at the MTRR A66G is that the minor form of that SNP, the GG form, all of those dots there, each one represents an astronaut. Everybody with the GG form of that SNP, the minor form of that SNP, developed ocular pathologies. And the fact that that cell in the bottom right is empty, this is around the time when some of the people that thought, I'm an idiot and that this is all crap, 2 different things, some of the people started to come around to thinking there might be something here. Now, this is not a smoking gun. This is not the one SNP you can go look at, because you can see there's a lot of people that developed eye problems that did not have that SNP. So it's got to be more complex than just one SNP, which we would have conceded that from the word go. The next set of data we came up with came from a bed rest study. This was a strict head-down tilt study that I won't belabor the details, but when we broke out the subjects by those that had the alleles that we were looking for in those 2 SNPs that I mentioned earlier, those that had the risk alleles developed significantly greater retinal thickening during bed rest than those that did not. And we now have a ground-based model where we can simulate the same retinal pathology that we see in astronauts on the ground. And that was a big deal, because up to that point, we'd not seen any changes in the eye terrestrially that mimicked what we were seeing in flight. But we now had both flight data and ground-based data documenting that there's a genetic predisposition for some astronauts to develop these ophthalmic issues. And I choose those words very carefully, and I read that sentence specifically because there's 2 different ways you can read the word some. One is that not all astronauts develop this problem. And you need to remember that, because a lot of people rush to the idea that, well, this was all related to you go into space flight, you don't have gravity holding the fluid down in your feet. The fluid all rushes up in your head, increases the pressure in your head. That pressure pushes on the optic nerve, pushes on your eye, and causes this problem. If everybody developed this problem, that would be absolutely true. The other thing we thought about was that carbon dioxide levels on board space station are about 10-fold higher than they are here on Earth. And again, as you all probably know better than me, when you expose people to high concentrations of CO2, it increases blood flow into the head, which everybody leapt on the idea that, well, it's the CO2 on space station which is causing blood flow into the head, which is causing pressure on the eye and pressure on the optic nerve and causing these problems. If everybody developed these problems, that would be true, but they don't. The other interpretation of the word some is that we maintain that genetics is a predisposing factor. It doesn't mean that everybody that does develop an eye problem in flight, it's because of their genetics. It's very plausible that there's more than one cause for this factor. So I'm not arrogant enough to tell you that this is it, but it is something that we need to look at harder to better understand it. One of the criticisms we faced after we came out with those data was, well, you know, how can your genetics and how can this vitamin stuff affect your eyes? So we sat down with the literature and we developed a hypothesis that is shown here in this figure, and it's actually, this is a time lapse, if you will, of the work that we've done. We came up with two different concepts, and that's not to mean that they're right. These are hypotheses, and as you know, hypotheses are starting points for research, not endpoints. On the right half of that graph, what it shows is the idea that your genetics can affect your B vitamin status, can affect nitric oxide synthesis and endothelial function, and that if your genetics drive you to have lower B vitamin status, your endothelium cannot work properly, which can lead to leakage, which can lead to edema, which in and around the optic nerve can cause a blockage of CSF drainage out of the head, which can push on your optic nerve, push on your eye, and potentially lead to these problems. That was the first hypothesis we came up with. We expanded that a couple years later with the left half of that image, which gets at the exact same thing, which is your genetics can affect your B vitamin status, can affect nitric oxide synthesis, that is, you'll have less nitric oxide and endothelial dysfunction, but it can also lead to oxidative stress. That is, it'll lead to increased peroxynitrate concentrations, which can stimulate matrix metalloproteinases, which can increase the turnover of the structural components of collagen and elastin. And that can happen anywhere in the body, but of interest here is that it can cause a decrease in the elasticity of the back of your eye, making it more susceptible to things like fluid shifts, which can occur in spaceflight. So again, we maintain that genetics and B vitamin status predispose some individuals to these pathologies. The multi-million-dollar question to this point is, can we counteract it? That is, if we were to give B vitamin supplements of the right forms, of the right vitamins, could we change the biochemistry to change either your nitric oxide production, endothelial function, and or to help build up the structures of the eye so that they're not as the elasticity is back to normal, and we are working on that. We had proposed a bed rest study that we managed to get funding for. The bed rest study started this week. Long story short, we were told about 5 months ago that because we were doing that study in Germany and because of the way the German laws are, literally we could not do that study because the folks at the German Space Agency that were supporting this study could not do the documentation required to meet the laws in Germany of any time you do a study where you're giving something to treat a disease, it falls into a different category of research. So the managers at NASA came to us and said, look, we need to pull your vitamin supplement from this countermeasure study. But we would like to move from bed rest and do this during flight. And right now they're off evaluating the proposal that we've rebuilt to go off and do this, to start giving astronauts these vitamins, which is something that frankly we should have done 8 years ago to see if we can counteract this problem. And hopefully if you invite me back in maybe 5 years, I'll have an answer for you. Now, some of this work has led to conversations with Dr. Moline about the idea that if we're talking about endothelial function and nitric oxide synthesis, you've got to get into the glycocalyx, which is an integral part of the endothelium. And as the image here shows, that the glycocalyx is, and you've heard a lot this morning about how important the glycocalyx is for endothelial function, and that when the glycocalyx is broken down, you end up with things like thrombosis and leakage through the endothelium, which again is one of the things we've posited as a possible problem with astronauts' eyes. When you start to talk about clot formation, you obviously not need to drag in Virchow's triad. And again, I'm sure everybody in this room is more familiar with this than I am, but this is a hallmark of blood clot formation and the different factors that are involved in this. And when we look at this during spaceflight, there are multiple factors during spaceflight which can impinge upon the three arms of Virchow's triad. That is, can affect hemodynamics, can affect blood coagulability, and that there's reason to believe that there's endothelial injury potential during spaceflight. This is another one of those things that we never thought we had a problem. Until a couple of years ago, during an experiment where they were looking at the internal jugular vein of an astronaut, people on the ground looking at that realized they had a blood clot that was sitting right there. And that woke a lot of people up. And you see there was an article written in the New England Journal of Medicine, and there have been some subsequent publications, and this has received a lot of attention and a lot of focus. I will tell you, this is the first time I'm about to utter these words in public, but we've got data, much in the same way we did with homocysteine, that help explain biochemically, plausibly, why that astronaut developed that blood clot. And that answer is oral contraceptives. Many, but not all, women astronauts will use oral contraceptives to suppress menstruation during flight so that they don't have to deal with that. Again, not all, but many. When we looked at oral contraceptive use, and everybody and their dog knows that oral contraceptive use increases blood clot risk. Okay? Some of the factors associated with that are that oral contraceptive use will increase transferrin. Oral contraceptive use will increase inflammatory markers like CRP. Oral contraceptive use will decrease serum albumin. We've got a manuscript in review right now showing all three of those things in the astronauts that used oral contraceptives to suppress menstruation during flight. And what we maintain is that oral contraceptive use leading to a lower concentration of albumin in the blood to the point where you would consider clinically hypoalbuminemia may be affecting the glycocalyx to the point where you're getting blood clot formation, and that is a significant concern for astronauts as well as obviously people on Earth. I come back to an image that I use a lot when I talk, and I make the point that when we talk about nutrition and space exploration, if we look at exploration on Earth through the history books, scurvy is a great example of how we learned the hard way about nutrition. And everybody has a vague idea that scurvy was a big deal, realized that scurvy killed more sailors in the age of sail than all other causes of death combined. And while it's interesting, perhaps more interesting, to study physiology or pathophysiology, we always need to remember that you need to get down into the grit of the biochemistry, and specifically the nutritional biochemistry, because lemons and limes didn't cure scurvy through magic, they cured them through biochemistry. I thank you for the time and the opportunity to present today. I need to point out the fact that while I got to come to Denver and to visit this great place and to visit with all of you people, there's a lot of people working hard behind the scenes. There's a lot of people around the world, literally, that have collaborated with us, including Dr. Moline, that have helped to bring this story forward in the last 10 years of work that we've been doing. And I leave you with an image of a book that we published earlier this year. We've published a few books over the years. The one in the middle there is our newest one. It came out a couple months ago. The URL at the bottom of the page is a site where you can actually download that book for free. And that book represents, in essence, everything I know. And we're giving it away for free. As I mentioned earlier, I work for the government, so I can't accept royalties. So you're welcome to have that book and have at it. The book on the right is one that we built for what I would call upper elementary or middle school kids. I would say it's the same as the other book. It's just got better pictures and smaller words. And that is available at that same website. You can download that as well. And with that, again, I thank you for the opportunity to present here today. And I would be happy to try to answer any questions. Thank you.